And as I have already announced, each participant please take 5 minutes for your presentation, and last 5 minutes we can discuss.
Okay, is it open
Yeah,
already?
yeah. Some I think again we have to. Somebody has to mute their.
మేడం స్లైడ్స్ కనిపిస్తున్నాయా మేడం స్లైడ్స్?
Yeah, yeah. I can see the slides. Please just zoom it, na.
ఓకే.
Yes, ma'am, visible.
Yeah. Full screen. You can use full screen.
ఓకే మేడం. Here the net is very slow, madam. Sorry for that. Sorry for that. So,
Don't talk to here. Madam, sir, please mute your mic.
shall I go? Maybe. Some network problem is there. We are able to hear, but slides are not moving, madam. I am trying to.
Now they are moving.
Okay. Okay, madam. Thank you.
Yes, yes. Somebody background. I think Dr. Nagaraju or who is that? Mute your mic.
Mute this one, sir.
Yeah.
Okay, madam.
Yes. Yes.
Respected HRDC, OU, Director Smita Power Madam, Course Coordinator Professor Madhavi Madam, today's resource person Karuna Madam, and my dear co-participants, a very good afternoon to one and all. Today we are going to present a talk on overview of sericulture and molecular identification of newly isolated bacterial pathogen from tussar silkworm. So our group consists of myself, Dr. V. Sridevi, Assistant Professor of Zoology from Government City College, Hyderabad, Dr. KLV Prasad Rao, Botany, Dr. D. Nagaraju, Assistant Professor of Botany, Dr. K. Anand, Assistant Professor of Zoology from Government City College, Hyderabad, and Dr. Kuntamalla Sujata, Department of Sericulture, Kakatiya University, Warangal. So now coming to introduction, what is sericulture, how it is important, and what are the types, and these are the things which we are going to discuss in my presentation. Sericulture is the cultivation of silk through rearing of silkworm for the production of silk. Sericulture is an alternate crop to tobacco, maize, etc., which give frequent returns to farmer in every alternate month by an income of rupees 35,000 to 45,000 per crop. Such crops can be harvested 4 to 5 times in a year per acre, so that the farmer can sustain his family economically. So, on an average, a farmer can generate an income of rupees 20,000 per month by this sericulture farming. So this is the farmer friendly industry, we can say, and also women also easily involved and handle easily in this silkworm farming. So we can say this sericulture industry as a woman friendly, farmer friendly, and eco friendly industry. And it is also we can invest less and generate more income through this industry. It is an agro-based cottage industry. It involves the raising of food plants for silkworm, rearing of silkworm for the production of cocoons, reeling and spinning of cocoon for production of yarn, etc. India is the second largest country in the world in producing silk after the China. Silk is known as queen of textile and biosteel because of its strength. Now coming to historical perspective of sericulture. According to a Chinese tale, uh, the Chinese Empress Li Ju has discovered the silkworm and silk production from the cocoon in 2640 BC. How it has been happened means At the time, she was walking in her garden and having tea and sitting under the tree and having her tea. Suddenly, a cocoon fell down into her teacup, and she was surprisingly observing that what is happening. Then she plucked it from her teacup. The cocoon unraveled and formed a beautiful string of silk. Then she looked up at the mulberry tree above, which had caterpillars crawling among the leaves. The empress concluded that the caterpillars were responsible for creating the fine silk. So soon after her observation, she taught this message to the stakeholders and it became widespread. Now coming to types of silk and its distribution. There are four types of natural silk produced around the world. They are mulberry silk, tropical tussar silk, temperate tussar silk, muga silk, eri silk, and oak silk, etc. And mulberry silk mostly concentrated at Europe, China, and USA. Tropical tussar silk concentrated at tropical forest zone ranging from Bihar, Jharkhand, and Karnataka. Temperate tussar silk is at sub-Himalayan region and eastern India. Muga silk at Brahmaputra Valley. Eri silk at Assam and eastern parts of India. Among them, the mulberry silk contributes around 90% of silk production, and the mulberry silkworm Bambyx mori is being regarded as the most important widely used species and extensively studied. These are some of the different types of silk moths. The first one is mulberry silk moth, that is light white color, and tussar silk moth is in lightish orange color, and muga silk moth is in brownish color, and eri silk moth is in grayish color. So now coming to life cycle of Bambyx mori, how the Bambyx mori forming cocoons. So the first stage of Bambyx mori's life cycle is egg. When the female lays eggs, it will hatch into a larva. The larva undergoes several instars. First instar, second instar, third instar, fourth instar, and finally the fifth instar. Uh, During that period, each instar will take 2 to 3 days for their development to reach the next stage. After completion of fifth instar, that will converted into pupa, and again the pupa develops into the adult form. This is about the life cycle of Bambyx mori. Same. Okay.
Yeah.
Now the stages of production. So sericulture can be divided into three divisions as follows. Cultivation of mulberry comes under agricultural division. Rearing of cocoons comes under entomological division, and reeling of cocoons comes under technical division. Now the next presentation will be continued by Dr. KLV Prasad Rao.
Thank you, madam. Thank you,
Over to
madam.
you, sir.
Sir Nagaraj, sir, will you present, sir?
Yeah, yeah, sir. I'm sharing the
Good
next.
afternoon.
This one. Stop అయినందా?
ఎక్కడికి పోయింది ఇది?
Share the slides first.
Now is it okay?
No, okay, sir. It's visible.
Yes, sir. Yes, sir. It is visible. So, good afternoon, Karuna madam, and good afternoon to all my friends. So, as a part of continuation of
Can I see you? Can I see you, Araprasad Garu? I
Yes, sir.
didn't see. I didn't see Sujata madam also.
Sorry, madam. Sorry, madam. Sure, madam. Since
Yeah, once you can come on screen and then you can
Good
come.
afternoon, ma'am. Good afternoon.
Yeah, good afternoon.
So I would like to continue the presentation with one more aspect which is important along with the sericulture, ma'am, because sericulture is dependent completely on the moriculture, which is about cultivation of the mulberry plant, that is Morus alba plant, and sir, first, can you go to next slide, sir? Morus alba plant. So it is dependent. Sericulture is dependent on moriculture, as I said earlier. Completely the feed of the mulberry, the feed of the silkworm is completely dependent on the mulberry plant leaves. And the first time mulberry plant cultivation was carried out, it was around 2800 BC by the Chinua. And the raw silk industry is completely based on moriculture and sericulture. And to increase the silk production and cocoon production, moriculture is an important process. So the moriculture technique have been improved with time and reduce the production cost in the sericulture. So next slide, sir. So approximately around 4 lakh hectares of farmland is used for the cultivation of the mulberry plants. And each hectare, approximately 12,000 to 15,000 kg of mulberry leaves can be obtained, which is using as a feed for the silkworms and rearing of it. And presently, the production of mulberry leaves has been increased due to the advanced techniques that are implementing in the cultivation of mulberry plants. And there are 4 main varieties of mulberry plants are being cultivated in India, especially. One is Morus alba, the second one is Morus indica, and the third one is Morus latifolia and Morus niagara. So next slide, sir. So I would briefly like to describe briefly about the optimum climatic conditions and what type of soils are required and what are the various irrigation procedures that are to be followed for the cultivation of the mulberry plant. Coming to the optimum climatic conditions, the mulberry plants generally require a temperature of approximately 24 to 28 degrees centigrade, even though it can thrive under a wide range of climatic conditions, but it is the suitable optimum temperature is 24 to 28 degrees centigrade. And although they give an average yield of at 28 to 24 degrees centigrade temperature as well, and it grows well in places and have an annual rainfall of approximately 600 to 2500 mm, and it requires around 350 square water every 10 days. And also the sunlight is a must for it, and approximately 9 to 13 hours of daytime period at direct sunlight should be required for the cultivation of the mulberry plants, ma'am. So next slide, sir. Next slide, sir. Next, coming to the soil conditions. The slide is not going, sir. Next slide. Coming to the
slide is not changing. Yeah. Okay.
Coming to the soil conditions, the most preferable soil for it is clay and the loamy soils, ma'am, and they are highly suitable for the mulberry plant cultivation. And the soil should be deep and it should be fertile and it should have a water holding capacity. Those are the prerequisites for the soils that are suitable for cultivation of the mulberry plant. And the ideal pH to be maintained, it should be at 6.2 to 6.8, that is slightly alkaline. And land preparation and for mulberry plant cultivation involves deep ploughing up to depth of 30 to 35, 350 meters. So coming to next slide. And also the requirement of spacing. The spacing between the plants should be the mulberry plants. It depends on the soil conditions again. And for the black and heavy soil type of conditions, it is 90 into 90 centimeter spacing is required. Whereas in the sloping soils, the hilly areas, it requires 220 into 60 centimeters spacing is required. And then the fertilizers and that are used for the moriculture exclusively, we generally use the sulfur fertilizer along with the biofertilizers also. So the biofertilizer also one more important fertilizer that is used for the cultivation of the mulberry plants. As there are some procedures like such as should mix biofertilizer with 50 grams of 50 kg of both types of like for example azospirillum and also the phosphate azospirillum and also the phosphobacterium. So all those biofertilizers are mixed in specific, sorry, previous years, previously biofertilizer. And also there is necessity of micronutrients also requirement is there for the plant. So micronutrients requirement is also there as uh, recommended for the micronutrients according to the beneficiary symptoms expressed, apply the micronutrients. And the wetting agent also TPOL should be added to it. So those are the requirements of the fertilizers. And coming to the different methods of irrigation that we follow for the mulberry plant cultivation, there are mainly three major types of. Sir, next slide, sir, please. Next slide. So there are three major methods of irrigation patterns to be followed for the cultivation of the mulberry plant. One is the ridges and furrows method. The second one is flat bed method, and the third one is drip irrigation method. So the first picture you can show, you can see the ridges and furrows method, whereas it is the most efficient method of irrigation and comparatively requires less amount of water. And the furrows serves as a irrigation channel. Irrigation channels are drainage channels during the heavy rainfall, so that the plant saplings cannot get affected with the heavy rainfalls and all. Coming to the second method of irrigation is the flat bed method, which is rectangular in shape. Generally, rectangular forms of beds will be prepared, and channels also formed, and the water is runoff will be relatively slow in comparison with the ridges and furrows method, and more land is wasted and requires more labor in the field of the field preparation in the case of flatbed method. And the third and the most important method, and which is a sustainable method, is drip irrigation method. So wherein the consumption of water is also less, and also it is very efficient. And the plant, the water in the form of droplets, in the form of drips, will be at the base of the plant, and even we can transfer the fertilizers and chemicals whatever we want to treat the plants. With the help of the drip channel itself, we can send those things also in the drip irrigation methodology. So these are the three methods. And coming to the next thing is fertilizers and preservations used. So the fertilizers are applied on the ground, the soil directly on a tree average of 7 to 10 days to ensure the optimal growth and development of the mulberry tree. And one more important factor that should be needed along with the weeding, the pruning process also should be done. So the pruning is the method will be conducted that to not to form the as a tree it should grow. So bushy form, to avoid the bushy form, the pruning will be done. But it is acceptable when the plant is grown in the pots. So the pruning process doesn't require. So the mulberry leaves should be kept after harvesting. The most important thing is for the as a feed of to use feed of as a feed of sericulture, silkworms. So the mulberry leaves should be kept in a basket full of water, and the basket should be lined with clean wet cotton cloth, and it should also be covered with a damp clean cloth. And if you want to store large number of mulberry leaves, we should use the storage rooms which have a moist green mulberry leaves, which are suitable for the feeding of silkworms. So the further regarding the diseases, what are the pests that are occurring to the mulberry plants, will be presented by my colleague Dr. Nagaraj sir. Sir Nagaraj sir, I hand it over to you.
Yeah. Yeah, sir. Okay, sir. I will share. I will stop it, sir, and just a second, please. Okay. Is it okay now?
Yes, sir. It is visible, sir. It is visible.
Yes, sir, visible.
Screen is visible.
Okay. Okay, sir.
Please carry.
Okay, sir. Good afternoon, ma'am.
Good afternoon.
Good afternoon, ma'am. This is Dr. Nagaraju, Assistant Professor Botany from Government City College. Now I'm going to discuss about the disease and pests of mulberry plants, which are affecting majorly to the mulberry crops. Is it moving, ma'am? Slides?
Yes, sir.
Okay, these are the whole sericulture process from the plant to pattu shari, how it is making the whole processes. Major diseases which are mulberry get affected with the fungi, bacteria, nematode, pests, not only one. Here the first fungal diseases which are leaf spot, that is caused by Cercospora maricola, and powdery mildew by Phylactina corella, and leaf rust by Ceratilium pisci, and sooty molds and mulberry stem canker by Lacediplodia theobroma, and leaf blight by Alternaria alternata, and stem canker by Botodiplodia theobroma, and cutting rot by Fusarium solani, and collar rot by Poma Sorgiana, and dieback by Botodiplodia theobromide, and root rot by Rhizoctonia botanicola, and bacterial disease also bacterial blight by Pseudomonas aeruginosa or Xanthomonas campestris, and nematode disease, root knot nematode disease, the Melodigene incognita, and most of the pests also cause the damage to the mulberry. That pink mealybug, the Maconilis irsites, and papaya mealybug, and Paracoccus marginatus, and leaf roller, that is Dipania pulverulentis, and Bihar hairy caterpillar, Spilactera obliqua, and thrips also, that is Pseudodendrothis mori, and white flies. These all are causing the damage to the mulberry. Yeah, the leaf spot, it is caused by Cercospora maricola. There is mainly the brownish necrotic spots, irregular spots appear on the leaf surface. The spot enlarges and it causes the damage to the whole leaf, so that whole leaf will be get damaged with this fungi. That is Cercospora maricola. Fungal picture also there here. And then the powdery mildew, the white powdery patches appear on the back side, lower surface of the leaf. That is caused by Pelactinia corallina. This also caused damage and widespread this disease with airborne, majorly airborne diseases. And leaf rust, that is circular pinhead sized brown eruptive lesions appear on the leaves. Later leaves totally become yellow or total wither up. These also spreading by airborne, whatever the fungal spores that spread from airborne. Major at temperature is the 20 to 26 are highly related humidity cause for this, and we have the some control measures for that. As already Varaprasa said, wide spacing of the plantation is needed. Pair row planting system is needed. Avoid delay of leaf harvests and spraying the chemicals. And sooty mold, also one of the major disease that having the thick black coated develops on the upper surface of the leaves. Totally black surface of like a smut. Totally it occupy the leaf space. It stop the lighting and photosynthesis. And majorly white flies in the mulberry peel, it caused the spreading of this sooty mold. And the stem canker, stem canker, it is caused by Lacediploidia theobroma or Andersonia torulidea. That also, when attacked with disease, inhibit the sprouting of the mulberry, and cutting leads to the iron mortality and black eruptions here and there, and black eruptions on the bark, and discoloration and drying of the stem is caused by this fungi with the stem canker disease. A total greenish black lesions on the stem. The total bark will be moved and removed from the. And then you can see the fungal leaf blights. These are caused by Alternaria alternata. They also airborne disease. Total leaf spots, it is totally blackening the leaves and the leaf margins, and it causes brown and it fall from the plant. Okay, the mainly temperature moisture is the main spreading of the disease, causing disease, and wide spacing is the more and more important for this all the control of all the diseases, fungal or bacterial diseases. And another one, the bacterial leaf blight caused by Pseudomonas aeruginosa. Uh, it is soil borne and seed airborne. Both the by both the ways it appear on the mulberry. Blackish brown irregular water soaked patches appear on that. And then the next is nursery, even in the nursery level also so many diseases before going to show plant, that is stem canker, that is caused by Botany theobromide, and cutting rot by Fusarium solani. The total dark bark will become the rutting and all cutting resulting in the death of whatever the sprout cuttings we use. And collar rot disease, that by Poma swargina, it brown or black discolorations of bark. And then the dieback disease is by Botryophylla theobromide, wilting from the tip to downwards, from the tip leaves to the downward total root, up to root also it damage. And some of the root diseases also caused by nematode disease, that is nematode is Meliodagne incognita, that caused stunted growth and with low water moisture in leaves and later yellowing the leaf margin. There is a nematode which cause disease, totally widespread in the by the soil borne. We can control this by neem cake oil, by spraying it neem cake oil during the interculture operations and after leaf harvest, making trenches at the near 10 to 15 centimeter deep near the root zone, and then plant soil by covering the soil with that. And next is root rot disease is caused by Rhizoctonia botanicola or Macropomena facilina. It's also not only single, they are associated with the other fungi also, Fusarium botanicola. And two type of the symptoms, above the ground and below the ground. It affect all plant. The above the ground, it is sudden withering of the plants, and leaves fall up from the bottom of the branches. And the below the ground, decaying of the root cortex. The root cortex or skin turn black due to the fungal spores. Affected plants after pruning fail to sprout, or plants sprout to be small or pale yellow and then with a rough surface. These are the Rhizoctonia botanicola that cause the disease, fungi. And there are so many factors that cause, especially the precautions we need to take for that is during the first 4 to 5 days, remove the dead mulberry plants, burn without exposing to the sunlight by out to widespread to stop the widespreading, and also 0.2% Navinia solutions we can use to control it, and whatever the organic contents. And then the next is pink mealybug. These all are the pests. Pests also cause so much of the damage to the mulberry. And pink mealybug, that is Maconelicus icitus, that is called the deformity. This is also called as tukra. Leaves become dark green and winkle thicken. Here you can see the dark green winkle and thicken, and bunchy top like appearances. Bunchy top like appearances is there. It also causes, especially the mechanical infection by the touching whatever it is, mechanical transformation is majorly. By biological control, Cryptolemus motorogene, by using this, we can control this insectivorous fungi. By using this, we can control these insects, mites, thrips. And you know, where is the insecticides, biocontrol agents. The papaya mealybug is, is a Paracoccus marginatus, an exotic pest. Here is the malformation of affected portions, stunted growth of leaf is caused. They are totally malformed, growth is stunted, and totally the presence of red and black caused by, especially the carried out by these insects. The insects, we can control this biologically by Asterophagus papaya, as a biocontrol agent, and also remove. We need to remove the whatever the secondary hosts, Partinium, Sida, Altinon, Getropa. By removing that, we can stop the spreading. And mulberry leaf roller, that is Diphonia pulverulentalis, that is the total whenever the leaf larva attack, the totally binds the mulberry leaf bundles. It turn it bundles by silken threads, stay inside and feed whatever the nutrient material. And by removing the pested portion, we can control it. And by spraying the DDVP or and then also by second spraying the neem, and also by using the Trichogamma colinus egg parasite fungi, that by using that we can control, we can stop, we can kill that leafy roller. And we have airy caterpillar, that is Spilatica obliqua. This also causes a very messy appearance. By this messy appearance is there. These also we can control by DDVP, by spraying it, and also by neem pesticides, azadirachtin, and by biological pest control by using the Trichogamma colinus, same for all the pests. It is a highly useful that used as a biocontrol, as a bioinsecticide. And not only fungi, bacteria, pests, thrips, insects also that causing the disease to the mulberry. That is Pseudodendropsis mori, that cause streak in early stages. The yellow brown blotches appear upon the leaf. After attacking this, the total whole plant will be become dry. And by using the chemical spray and biological control by predatory ladybird beetles releasing, we also can stop spreading of this disease. And then white fly is the major causing the spreading transmission of the disease in the mulberry, especially that transfer the disease from one to other by the spiraling of waxy material is the typical symptom here. This also we can control by use sprinkler irrigation, as Sir said earlier, drip irrigation or sprinkler irrigation. We can some more control than the normal irrigation. And chemical control by spraying DDVP, and biological control by Cryptolimus monitor by using that. Also Simonus cucubora, by using this we can control adult beetles, whatever it is, we can prevent from this. These are the old diseases which cause damage. By taking care of it by using the biocontrol method, by using the advanced techniques, by these we can control this all the diseases with minimal exposure, minimal knowledge. Also we can control these diseases. Our next presentation will be given by Anand sir. Please, Anand sir.
Sir, yes, sir, I am online.
Yes, Anand, go ahead.
Good afternoon, ma'am.
Good afternoon.
Good afternoon, Dr. Karna, ma'am, and my co-participant. I am Dr. K. Anand, Assistant Professor of Zoology, Government City College, Hyderabad. Today, if the slide is visible, yeah.
Yeah, go ahead, Anand.
Yes, ma'am. I am going to present about the production of silk. So the major part of the sericulture is the production of the silk. It mainly includes the rearing, stifling, and rearing methods. So finally, we will get the silk products. And rearing of the silkworm. The slide is moving.
No,
No, sir, no, sir.
slide did not change.
Okay.
Go to the presentation, sir.
Yeah.
Okay, now it is okay.
Yes, sir. Now it is.
Okay, sir.
But we want full screen.
Yes,
Please
ma'am.
make it full screen.
Yes, ma'am.
Sir, at bottom you having the arrow mark.
Yes, sir. Yes, sir. I'm doing. Yes, sir.
Okay, sir, continue.
Okay, sir. So in the rearing of the silkworm, in the beginning, the female silk moth lays hundreds of eggs These eggs are stored over a clean paper or a piece of cloth. These eggs are then sold to the farmers. The farmers then keep the eggs under the accurate temperature and humidity in a clean place. They are warmed to the most appropriate temperature to hatch the eggs to the produced larvae or caterpillars. This process is done when the mulberry trees have a fresh crop of leaves. The caterpillar eats these mulberry leaves day and night, and it grows in size. And bamboo trays are used to keep the caterpillars, and some freshly chopped mulberry leaves are kept in the tray. After 30 to 40 days approximately, the caterpillars stop eating the leaves and then moves inside the small chambers in bamboo trays to spin cocoons. These are produced by the secretion of the liquid protein from their salivary glands. Small racks are given in the trays so that the cocoons attached to those racks. The silk moths are developed inside the cocoons.
Sir, slide is not moving. You go to
Yeah,
presentation,
yes, sir.
sir.
So it was structured. Sorry for the inconvenience.
Okay, now it's moving.
Okay, sir.
Next slide. Third slide. Yeah.
So in this rearing process, basically, it is two types: chalky rearing and the late age rearing. So in the chalky rearing, the hatching to its full grown stage, the silkworms pass through the 5 instars, and the worms up to the stage 2 are called as the young age worms or the chalky worms, as they are susceptible to infections and vulnerable to adverse weather and other conditions. Special care is required for the rearing of the chalky rearing. In the later age rearing, rearing of late age worms begin from the third instar. These worms are voracious feeders, and it requires the rearing house and rearing appliances. Mulberry silkworm rearing being completely domesticated and demands the specified environmental conditions like temperature up to 24 to 28 degrees and relative humidity 70 to 85. It is therefore necessary to evolve for economic cooling through the selection of proper material for wall and roof fabrication, orientation of the building construction method, and the design, etc. Further, enough space must be available to carry the leaf preservation, chalky rearing, late age rearing, and molting. It is also convenient enough to conduct effective cleaning and disinfection. The size of the rearing house depends upon the quantum and type of the rearing. A floor area of 400 square feet can provide the rearing space for 100 DFLs, that means disease-free layings. So 1 DFL contains 500 larvae. And the rearing appliances, the latest silkworms do not tolerate high temperature, high humidity, and poor ventilation. Hence, the rearing house should have cross ventilation facilities to bring down the room temperature and for removal of vapor and harmful gases generated from the large quantities of excreta produced by the silkworms. The minimum requirement of equipment for 100 DFLs larva is given in the table in the next slide. So according to this, these are the minimum requirements. Shoot rearing rack 40 by 5 inch. They are the 5 tires, one quantity. And rotary mountages are the chandrikas, so 35. And power sprayers, one power sprayer, hygrometer, along with that one chopsticks and a rearing tray and feeding stand, all these are required. And feeding, maintenance of temperature and humidity is very crucial in the silkworm rearing. The initiate feeding with 50 to 55 days old shoots harvested at the height of 3 to 4 feet in the cooler hours of the day, preferably in the morning. 60 to 65 days old shoots should be fed to fifth age worms. The harvested should be preserved loosely in the vertical position in cool and moist place by covering with cleaned, disinfected, and wet gunny cloth. The quantum of mulberry shoot required is 460 kg in 4th instar larvae and 2808 kg in 5th instar for bivoltane silkworms. So usually daily 3 feeds with the intervals of 6 a.m., 2 p.m. and 10 p.m. schedule should be followed. The ideal temperature for the late age is 26 degrees centigrade for 3rd instar larvae and 25 degrees centigrade for the 4th instar larvae and 24 degrees centigrade for the 5th instar larvae. 80% of humidity is required for the 3rd instar larvae and 70% humidity is required for the 4th and 5th instar larvae. And harvesting and sorting of the cocoons and the yield of the cocoon and marketing is the further stages in the production of the silk. In the harvesting and shooting, the harvest cocoons on 6th day, remove the defective cocoons. So by removing of that one, after the sorting of the defective cocoons, these are graded into the cocoons according to their quality. In winter, delaying the harvest by 1 day will be applicable. And cocoon yielding is the average yield is 60 to 70 kg from the 100 DFLs. About 700 to 900 kg cocoon can be harvested from 1 acre of mulberry garden in a year. And marketing, the transport of the cocoon during the cooler hours of the day and market on the 7th day. The cocoons need to be loosely packed in nylon netted bags up to 30 to 40 kg capacity and transported in vehicle having shelves or the partitions so that pressing of the cocoons can be avoided. And processing of the silk from the cocoons. So this is by the major processing, stifling, reeling. These are the major processes for the production of the raw silk thread. So in the processing of the stifling, the cocoons are used to obtain silk threads. When the cocoons are exposed to the sun or steam or boiled, the silk fiber gets separated. And reeling, the process of getting the silk threads from the cocoons to use as a silk fabric is known as the reeling of the silk. Reeling of the silk is carried out by the special machines. These machines unwind the fibers of silk or threads from the cocoons. Usually in the traditional way, the charkas are used for that one. The silk fibers are then converted into silk threads to make the different kinds of the silk fabrics like saris and other silk by the weavers, silk products by the weavers. So these are the this picture depicts about the stifling and reeling. And finally, the production of the silk yarn after dyeing different colors added to that one. Then by the weavers or by the machinery, they are weaved with the different types of the silk products, particularly in our area, Pochampalli, Gadwal, Narayanpet silk, Dharmavaram silk, and different types of the designs, Pochampalli, Ikat from Orissa. Those designs are specifically produced for the silk productions. So next topic will be proceeded by the Dr. K. Sujata ma'am about the pathogenicity in the silkworms and specified a pathogenicity with molecular basis from his work. Please carry on, Sujata ma'am. Thank you.
Sir, please close the slides, sir, please.
Okay. Yeah.
Okay. Good evening, madam. I'm Dr. Kuntamala Sujata, presenting my topic entitled Molecular Identification of Newly Isolated Bacillus paramycoides from tussar silkworms. This is in continuation to my other group friends. This is a part of the research work carried out by my research scholar Prashanti on tussar silkworm that was reared outside. That is, we go for outdooring. Introduction: silkworm diseases that are common in India are grassery, flatchery, muscardine, and pebrine. Of these diseases, bacterial flatchery in tussar silkworm accounts for 15 to 18%. The bacterial diseases are classified as bacteria septicemia, bacterial toxicosis, and bacterial gastroenteritic. Of these, I am concentrating on bacterial gastroenteritic, which is pronounced in larval period, causing a loss of 10 to 15% to the farmers. From the DFS that was rare in the sericulture farm, silkworm cadavers were collected on basis of the symptoms. That is bacterial symptoms. One is the chained soft pellet, scaled annual lip, shortened and brownish or body. This was carried out for the further studies. Then we went for the isolation of bacteria from the silkworms. From the gut tissue of the sterilized silkworms that were collected from the dissected silkworms, which we took the gut tissue, we went for the grinding, then we centrifuged at 3000 RPM, and the crude source of infection was formed. Then we went for preparation of the primary culture using streak plate technique. Using serial dilution, agar plating method, single colonies of the bacteria was isolated. The bacteria were cultured in the liquid broth, which were sent for molecular identification to NCBI. Today, 16S rRNA approach is a powerful tool used for identification. The bacterial 16S rRNA gene was amplified using polymerase chain reaction, that is the PCR. Then we purified using exonuclease 1 shrimp. Then the purified amplification were sequenced by genetic analysis and then edited using a chromoslide version 1.5. The data that was saved was analyzed by BLAST with closest sequence retrieved from NCBI program database and converted into next format final tree. Here for the isolation and molecular identification of the bacteria. On the basis of the multiple sequence alignment and phylogenetic analysis of 16S rRNA sequence, the strain was identified as SCQ 179 underscore SEZ0001 NC 220319, which exhibited high level of similarity with the known sequence in the public database NCBI and blast results. The bacterial pathogen is identified as Bacillus paramycoides. This is the figure is showing the sequencing partial 16S gene. This slide shows the phylogenetic relationship. Finally, it was found that the sequence of the closely related Bacillus species, when matched under multiple sequence alignment and phylogenetic analysis, showed that the tussar bacterial strain was under the group of Bacillus species and the strain showed closest homology with Bacillus paramycoides. This was the new pathogen which we discovered while we were going for the tussar silkworm rearing in our lab. These are the references, and this was a research paper which we have published. Thank you, ma'am.
Presentation is over.
Yeah, yes, ma'am.
Please come online.
Yes, ma'am.
Yes, ma'am.
Just a minute, ma'am.
Yeah. Sridevi?
Yes, ma'am.
Sridevi is there?
Yes, ma'am.
Okay.
Yes, ma'am.
Yes. First of all, I congratulate you. It was the topic chosen was really good, very informative, and I think commercial
Thank you,
point
ma'am.
of view, and what it is needed for the growth of our country. Definitely, a very good
Thank you, ma'am.
topic was chosen.
Thank you,
I
ma'am.
congratulate all of you,
Thank
and
you,
well
ma'am.
presented also. The
Thank you
amount
very
of
much, ma'am.
literature presented,
Thank you,
etc., was
madam.
really good.
Thank you, madam.
So first
Thank you.
speaker was Sridevi, right?
Yes, ma'am.
Yes, ma'am.
So I have one question, Sridevi, for you and the group also.
Ma'am.
Any genetically modified silkworms have been produced in the aspect genetically modified means you can target anything like more yield of silk or
Sujata
disease
madam.
resistant silk,
Sujata
etc.
madam. Sujata madam.
Yeah, any genetical
Sujata, madam,
modified
can
silkworms?
you?
Uh, yes, ma'am. Genetically modified. That is fluorescent. We have the fluorescent cocoons also. It is produced through the genetically modified silkworm, ma'am.
Okay.
And it so happens that you get colored cocoons. And it so happens that in rearing, we go for the multivoltine cocoons are colored. So we have to go for bleaching and then we have to go for the reeling, madam. But due to the genetic engineering, it was found that we have got the fluorescent cocoons where we can directly go for reeling of the silk from these cocoons directly, ma'am.
That's good. That's
And
good.
genetically modified hybrid silkworms also generated,
Yeah,
madam.
yeah.
They for the spider silk and this mulberry silk.
Yeah.
That mixture will be
We went
produced.
for,
Yeah,
you mean to say we
A
have gone for
combination of
crossing
both.
of
yeah, hybrid. Yes,
spider
ma'am.
and
Yeah.
also
Hybrid material
Hybrid.
produced by that.
So, so then one more question I have. Anybody can answer this. What is the chemical or biochemical composition of this silk?
it is sericin and fibroin, mat. That
Yeah.
is in the protein. It is a proteinaceous fiber, and
Initially.
we have sericin and fibroin. That sericin is nearly 22 to 24%, as well as fibroin is 72 to 80%, mat.
Yeah, and this sericin will be richly containing serine and glycine, two
Yeah,
major
ma'am.
amino
Yeah,
acids.
two
Yeah,
amino
yeah,
That
acids.
is the reason the name sericin has come for this.
yeah.
One question for Varaprasad Garu,
Sir, ma'am.
Dr. Varaprasad.
Yes, ma'am. Yes, ma'am.
Uh, what is the role of government and in this rearing and promoting this sericulture?
I mean, regarding
Having enough government support?
uh, not so much, ma'am. Not so much. Definitely, the farmers need to be encouraged in various ways by giving subsidies and all. There is lot of scope is there, ma'am, to help the farmers. It is not completely satisfied if you compare the farmer of the mulberry plant farmer with other crops, ma'am. Even though it is a cash crop, but we are not getting that much revenue in comparison with other other crops. And also incentives wise also I didn't find any kind of incentive from our state. For example, if you take the state of Telangana, there is no such incentives for mulberry farmers, the mulberry plant farmers. So it
Yes.
will be better if some kind of initiative from the government also, it will be encouraging for them, and also the production will be increased, ma'am.
Because we are having many sericulture, horticulture research institutes, it can be extended. The support can be extended in this form, right?
Yes, ma'am. A lot of awareness also needed, madam, for the
What is
government.
the lifetime of this mulberry plant?
I mean, after once you sow, ma'am, it will for complete. It's a deciduous plant, ma'am. For 6 to 7, 8 years, it will grow as an adult plant, ma'am.
Oh, 8 years.
It will grow as an adult plant.
So once you start growing,
Then,
7 to 8 years, the land will be occupied by this mulberry.
yes, ma'am. Lot of
And I just
lot
want to
of.
add, ma'am, I just want to add here, ma'am, just to, for, suppose you want to carry out rearing, suppose you go for the cutting method. Suppose we want to establish a mulberry garden, we go through the cutting method, ma'am. Just in 6 months, you can get a crop. Then after that, every 2 months, you get a crop, madam. That is, you can go for rearing every 2 months. Meaning to say, the silkworm larval instar or larval duration, it lasts for 28 to 30 days, depending upon climatic conditions and races. So because during that you get a complaint. And here I can add one thing is, in other agriculture crops, there were suicidal cases, but in sericulture till now, there is not a single case now. That is the greatness of sericulture, and that is why we have chosen this topic also, madam. Thank you very much.
Yeah, that's true. And what is the composition of these mulberry leaves? Is it itself rich in protein?
Yes, ma'am. There is a protein. Morin is a
Protein.
what we say, that is a
Major.
protein enzyme which is present in the mulberry leaves, madam. Because of that, the thing is, we can, because of the morin substance, we can we are feeding the mulberry leaves to the silkworm.
Yeah. So while I'm going through the different type of microbial and pest infections,
Yeah.
so I had one technical thing. This silkworm itself is one pest on that, right? We can consider
No,
it
ma'am.
as a
It's
pest.
not a pest. It's not a
It's
pest,
not a
madam.
pest.
It's not a
It's
pest.
not a natural pest.
It's not a pest.
So unless you feed it, it won't.
No,
It
as
won't.
such,
We
we
are
have madam,
domesticated
that's why
and commercially we
sericulture
are seeing,
puts
ma'am.
into.
Actually, in the pattern, it's feeding on the mulberry plant. So in the early stages, that may be the best, but it was evolved as a beneficiary product to the human mankind.
As such, we don't have only mulberry, madam. We have in non-mulberry, we have, as Sridevi madam has mentioned, we have the tassar, eri, muga, and oak tassar.
Oak silk.
So,
So one question to Dr. Anand sir.
yes, ma'am.
If you are going for sericulture,
Yeah.
under like in laboratory conditions, okay, what type of infrastructure you require for storage and for growth and maintenance?
So first thing we require the BOD, madam, and maintenance of the temperature and humidity, and then we are having those egg laying components, and chandrikas also we require for the rearing of that one.
Yeah,
And
because
we
in
require the
Hyderabad,
nearby.
quite few centers are there. Some private agencies are doing sericulture, right? In
Yes, yes, ma'am.
Hyderabad.
Private is also doing, and from the government side also, we are having the different centers. From in the near to Shadnagar, Kamadanam, there was a sericulture center is there, ma'am. That is one of that near to Hyderabad, and some private centers are there.
How about you people hold a workshop, sir?
Sure, ma'am.
It's a good
Then
team.
we introduced sericulture as a paper in our UG course, ma'am.
Right, right. If you can
Yeah.
hold a workshop, it's not just for educating, but you'll help a lot of unemployed people with the scope behind that.
Yes, yes,
Ma'am,
ma'am.
we went for a workshop, madam. Workshop
Yeah.
at university. Yeah, at our university also, we went for a workshop, madam. And we have called.
Actually,
Yeah.
we planned for a certificate course, madam, in sericulture.
Dr. Nagaraj sir, maybe
Yes, ma'am. Yes,
I
ma'am.
didn't
Yes,
touch
ma'am.
with you.
Ma'am.
Is there any treatment for these bacterial infections to avoid the bacterial infections in this silkworms?
Yeah, ma'am. Bioinsecticides are there in between. Now, instead of chemical pesticides, we use biopesticides.
Latest, we are identifying or coming to know about antimicrobial peptides.
Yeah, ma'am.
So, how far they are applicable, or are they in the applications for sericulture? Antimicrobial peptides.
Yeah, ma'am. This will be useful. Definitely,
Because antimicrobial peptides themselves are
protein,
isolated from various insects, etc.
insects. Yes, but that are even though it insecticidal capability is there.
The sericulture, the silkworm itself may have some AMPs also.
But
Maybe we need to identify.
yeah, ma'am. But it is not true. This it is a symbiotic. It is not damaging the insect, especially this.
Okay, sir and madams, nice presentation.
Thank you,
Thank you,
madam.
ma'am.
Thank you.
Thank you, madam. Thank you very much,
Yes,
madam.
yes. Good. I want to take a 15 minutes break here. Then we'll move ahead with the next group.
Okay, ma'am. Thank
Okay,
you.
ma'am.
Yeah? Next group, third group. Right?